The concept was all about the blood groups. Now I said that the blood group concepts arises because of specific molecules which are known as agglutinogens that is present on the cell membrane, the surface of the cell membrane, rather cell membrane of RBC. RBC key jinish I have already explained yesterday. Aro jodi jante chao what actually is RBC, you will be better viewing the old videos. Anyways, so these are specific proteins. In fact, antigens and the antibodies are also specific proteins. Proteins constitute, one of the very important molecules present inside our body. It's a class of molecules present inside our body. So agglutinogens are proteins that are present on the surface of the cell membrane. Now, suppose say this is an RBC, and if this be an RBC, then outside this layer is to be considered as the cell membrane, right? Now, on this cell membrane, there are
বিটান জয়েন হতে পারছে না, বিটান জয়েন হতে পারছে না,
বিটান জয়েন হতে পারছে না। সো কি করা যায়? ওকে, বিটান ইজ দ্য ওয়েটিং রুম। So on the surface of the cell membrane, there will be multiple proteins, and these proteins can have different labels. Labels bolte ekta A type proteins or A agglutinogens and a B agglutinogens. Clear? Now, if there be an agglutinogen, in that case, the specific type of blood having the A agglutinogen would be having an anti-A agglutinin. Let me repeat. The A agglutinogen comes with anti-A agglutinin. So this is the composition of the blood group that we call as A. Is the concept clear? এটুকু আপনি বুঝলেন যে যেটাকে আমি A anti A গ্রুপের ব্লাড বলছি, that particular blood is having A agglutinogen and anti-A agglutinin in that blood. Similarly, the particular group that I am calling as B would be having this B agglutinogen, and in addition to that, there is anti-B agglutinin. So now this entire thing is a A group blood, and the other here is a B group blood. What will happen if these two group gets mixed? Duto group ke jodi if we are mixing kore di, so what is the result? The result is that agglutinogen would cross react with the opposite agglutinin. এদের মধ্যে একটা ক্রস রিঅ্যাকশন হবে। তার মানেটা গিয়ে কি দাঁড়াচ্ছে? ধরা যাক আমি যে ব্লাডটা দিয়েছি সেটা A, সুতরাং আমার যে আরবিসিস গুলো এখানে সব রয়েছে এগুলো সবই A আরবিসিস রয়েছে। যতক্ষণ A আরবিসি রয়েছে তার মানে এই A এর রেসপন্সে একটা অ্যান্টি A, আমি সেটাকে অন্য একটা কালি দিয়ে দেখাচ্ছি, দিস রিপ্রেজেন্টস দ্য অ্যাগ্লুটিনিন, সেটা স্ক্যাটার হয়ে রয়েছে। ফাইন, এটার মধ্যে if I am introducing another blood, say that blood is of B, তাহলে কি হচ্ছে? With the blood B, what is entering out here is the agglutinogen that is known as anti-B, right? So because B is having anti-B and agglutinin. So in the positions of these blue colored dots will come now this, what color would I mention this of? This particular color dots, which represents, what does it represents? Anti-B agglutinin. Now here comes the problem. What exactly is the problem? This A antigen would bind to the anti-B. This A antigen would also bind to the anti-B. This would also bind to the anti-B. So instead of being in this scattered form, the form that we can now see is that all the A grouped RBCs are all clumped together, accumulated together in a big mass. And this mechanism is known as agglutination. Did you understand what is agglutination?
So please explain once again.
Here I have used 3 different colors. This particular situation is representing an A blood group. Representing an A blood group. So in the A blood group, what we are having is the A agglutinogens. Got it? And with the A agglutinogens, we are also having agglutinins, which are anti-A. So these are anti-A. With this blue color, I am showing you the anti-antigens because just now to the side, I have explained the composition of the A blood group. Have a look that this A group consists of what? A agglutinogen and anti-A agglutinin. That comprises the entire blood group, A group. Okay. So if I picturize that, I am having this A agglutinogens. These are present on the RBCs, that is the cells of the blood. And in the plasma of that blood, we are having in these blue colored dots the anti-A agglutinins. So as long as anti-A and A are present together, the individual cells remain scattered as if the fluid can flow, the blood would flow. The very moment when this anti-B agglutinin enters into this system, this anti-B starts reacting with the agglutinogen A present on the different RBCs, present on the different red blood cells. Okay. So what happens is a kind of a cross reaction. And that cross reaction makes all the cells come together the way I have shown it out here. So once solid cells come all together in a single space, so in one location you get a now big mass. That is what I've written. A big mass is now formed. Now, if in the circulatory pathway, suppose there is a blood carrying channel, let us consider that this is an artery, which is having a very definite size lumen. Lumen means the space in the artery. If this big mass comes in this space, would not there be an obstruction to the flow? If I place a big mass out here, suppose say this same system, I mean all the RBCs grouping together, if this system makes an appearance, something like this, I'm drawing the same thing out here. So would it not cause an obstruction to the flow of blood? This artery is the channel, is the pipe, you can say it that way, is the pipe through which the blood is supposed to flow. Now, inside that artery, if there is a material that is blocking the free space, would the blood now flow? Can the blood flow through an obstructed channel?
No, sir.
Since the blood cannot flow, that means what? The distant locations out here where I'm putting the dots, this location would not have any more further supply of the blood. If the blood doesn't flow to this location, that means the tissues present is not receiving any blood. And as the tissues present are not receiving any blood, the resultant consequence is that the tissue will gradually die. Is the concept clear now? Why tissue dies if we are putting an unmatched blood transfused? Understood? Now, this can occur in multiple different locations of the body, because multiple different locations of the body do survive on blood. And if blood fails to reach to the multiple different location, then the survival is now put into a question, right? Survival is now a doubtful issue. Are you getting me? Why it is always necessary to have this blood group matched? Because an unmatched would cause this phenomena, the phenomena of agglutination. This one. This phenomenon is known as what? I have written it somewhere, but I cannot see it anymore. Anyways, I have written it again. Agglutination. Is the concept clear? The agglutination is causing the obstruction, and the obstruction is causing the tissue death. So if I draw it in the form of a flow chart, what will be the flow chart? Unmatched blood. What does that cause? That cause agglutination. That is a technical word. In English, we also call it as clumping. CLUMP, clumping of RBC. So once this agglutinated mass cause obstruction, or technically it is known as occlusion. So I will write first the technical term. Occlusion or obstruction in blood flow. And since blood flow is getting impeded, is not flowing properly, so the tissue death occurs. And this can occur across multiple different organs. So that is how each individual organs do suffer damage. Have a look on the board and confirm whether you have understood this part or not. And then I will teach you the other blood groups as well. I mean, the other groups of this ABO system. কোনো প্রশ্ন থাকলে করে ভালো শর্টকাট। So if we are speaking of the blood group type, let me draw a table out here. Okay, red blood cell types based on the agglutinogen that is present. So we can write as A. We can write another type as B. So if this be the sera is the plural of serum. Yesterday I explained what is serum. Serum আমি আজকে বোঝাবো না, যারা বুঝতে পারছো না তারা আগেকার ভিডিও দেখে নেবে। Okay, so serum. So in there basically are two types of sera. One is anti-A and another is anti-B. Now যেখানে যেখানে A agglutinogen থাকবে, সেখানে যে সেরাটা থাকবে সেটা হচ্ছে অ্যান্টি এ অ্যাগ্লুটিনিন থাকবে সেরামের মধ্যে, রাইট? তো যেখানে অ্যান্টি এ থাকছে সেখানে অবভিয়াসলি আর অ্যান্টি বিটা থাকবে না, সুতরাং না থাকাটাকে আমি মাইনাস সাইন দিয়ে ইন্ডিকেট করছি। কনভার্সলি যেখানে আমার ব্লাড গ্রুপ বি থাকছে, মানে আরবিসির মধ্যে যেখানে বি অ্যাগ্লুটিনোজেন থাকছে, সেখানে আমার অ্যান্টি এটা থাকবে না, না থাকাটাকে আবার মাইনাস দিয়ে ইন্ডিকেট করছি, বাট ব্লাড গ্রুপ বি এর মধ্যে অ্যান্টি বিটা থাকছে। Clear?
Yes, sir.
Okay. আর ও এর মধ্যে ও আর একটা গ্রুপ আছে, তার আগে এ বি বলে একটা গ্রুপ আছে, সেটা বলে দিই। এ বি, এই এ বিটার মধ্যে যেহেতু তোমার এ রয়েছে, এ অ্যাগ্লুটিনের জন্য রয়েছে আর বি অ্যাগ্লুটিনের জন্য রয়েছে, সেহেতু এখানে তোমার কোনো অ্যাগ্লুটিনিন নেই। আবার ও এর মধ্যে, ও মানে কি এমটি, তার মধ্যে কোনো অ্যাগ্লুটিনোজেন নেই, তার মানেটা কি দুটো ধরনের অ্যাগ্লুটিনিন এর মধ্যে রয়েছে। ক্লিয়ার? দিস ইজ দ্য চার্ট।
Yes, sir.
অ্যাকচুয়ালি এই যে দুটো জিনিস দেখালাম, on the first two rows, this symbols here is representing the presence or absence of agglutination. Okay, presence or absence of agglutination. Now, what is that? When there is a plus sign given, that means there would be an agglutination. And when there is a minus sign given, then there is no presence of agglutination. Okay. So, AB when given, that means there would be no agglutination present. So that means what? AB can be considered as the universal recipient blood. Now, what is the concept of a recipient? That concept comes with the concept of the blood transfusion. And accordingly, O blood group represents the universal donor. Now, what is the concept of a recipient and a donor? See,
স্যার রেসিপিয়েন্ট মানে রিসিভ করতে পারবে।
exactly so. In blood transfusion, there is one party which will give the blood. এই যে বিভিন্ন blood donation camps গুলো হয়, here we actually donate the blood, and if we are donating blood, we are then considered as donor. In the camps we can have only the donors, but the patient who are hospitalized, who are in need of blood, তারা ব্লাডটা দেওয়া হবে, so they will receive. So that's why patients, donors can be the normal person, but the patients are usually the recipients. এখন whether the person donating or a person being a patient is receiving, whatever it be, the person would be having blood. Blood ছাড়া তো কোনো persons হয় না. So since the person would be having blood, the person definitely would be having only a particular type of blood of these 4 different types. Okay, চারটের মধ্যে যে কোনো একটা টাইপের ব্লাডই তার মধ্যে থাকবে। তো when I am getting a recipient with A, in that case I have to be very particular that the blood that recipient A receives would either be A blood group or be the O blood group. Why? Because O is the universal donor. O সবাইকে দিতে পারে, সেহেতু O can donate to all these 3 different blood groups. Okay, but if the blood group is A, in that case either O or A. There should not be any kind of B, neither there should be. There will be an AB. If these two blood groups are given, so I will mark it as negative. That means this shall not to be given. It can cause what? Agglutination, which would ultimately lead to death. Clear? Why only A to be given to a recipient who is A, or O can also be given to the recipient who is A. Clear? Following that
Yes, sir.
same logic, if B is the one group that the recipient is having, so for that particular group, you will have to give either this B or you can give O, but you can never give A. This is not done. This is negative. Because if you give A to a blood group which is having, if you donate A group blood to a recipient which is having B group blood, in that case, there again will be agglutination and the patient will die. Clear? Okay.
Yes, sir.
Now, if there is an AB, consider that this AB is having both the types of agglutinogen, but is having no types of agglutinine. So since it is having no types of agglutinine, there is no chance of the cross reactions. So it can receive blood from B, from A, as well as from O. So since it can obviously receive blood from AB as well. So since it can receive blood from all the 4 other different types of blood group, this is considered as the universal recipient. Clear? And
Yes,
O,
sir.
since it is not having any type of agglutinogen, in that case, it is only having both the types of agglutinin. So it can be given to any different types of, blood. So that's why this is known as the universal donor. Okay, so have a check on the board and find out whether there is any questions or not. Meanwhile, I will pause the recording again.